Good afternoon, guys. In this lecture, we will be discussing about the essentials of development and development of human face and developmental defects. Firstly, we will be discussing about the prenatal development. Prenatal development, it comprises 10 lunar months, and those months are of 28 days each. Well, this prenatal development is divided into three successive phases. The period of ovum, which lasts only for 1 week. Then the embryonic period that lasts from the 2nd week to the 8th week, and that period is divided into pre-somite period, somite period, and the post-somite period. And then comes the period of fetus, that is the 3rd period, and that starts from the 3rd month and ends on the 10th month. So over here, if we look at the progression, a fertilized egg becomes a zygote, and then it goes into the 2-cell stage, then it goes into the 4-cell stage, then it goes into the 16-cell stage, then it becomes a morula, and after that it becomes blastocyst, and then it occupies the shape, or it gets the shape of an embryo, and finally the child is born. The mating of male and female gametes in the maternal uterine tube initiates the development of a large totipotent cell, which can give rise to cell of all type, and having 100 millimeter of diameter, we call it zygote. Morula, the zygote, it undergoes rapid mitotic division to form the morula, having 16 pluripotent cells which can give rise to a specialized form of tissue. It has an inner cell mass which gives rise to embryo proper, and that is known as embryoblast. The outer layer of cells are trophoblasts, which help to provide nutrition to the embryo. Well, on the 4th or 5th day after conception, the fluid passes from the uterine cavity into morula and partially separates the inner cell mass from the trophoblast. The morula now becomes a blastocyst, and the cavity is known as blastocele. Placenta. On the 7th day, the zygote passes through uterine tube and implants into the uterine wall. The chorion develops from blastocyst by growth of villi into surrounding maternal uterine wall to tap the blood supply. The surface directed villi eventually atrophy, while basally directed villi, they are hytrophied and combine with uterine wall to form the placenta. Bilaminar germ disc. Some cells of the inner cell mass differentiate into flattened cells which constitute endoderm, the first germ layer. The remaining cells of the inner cell mass become columnar and form the ectoderm, the second germ layer. At this stage, the embryo is in the form of a disc having 2 layers. Developing placenta and the bilaminar germ disc. This is the developing placenta. This is the amniotic cavity, ectoderm, endoderm. Then we have precaudal plate over here. This is the secondary yolk cell. This is the uterine wall. Amniotic cavity. The space between the ectoderm below and the trophoblast above is amniotic cavity filled with amniotic fluid. Yolk sac. The space between ectoderm above and trophoblast below is yolk sac. The fluid of amniotic cavity and yolk sac is meant for protection, nutritional exchange, and waste elimination. Here we can see from the egg, then fertilization, fertilized egg, then 2-cell stage, then the 4-cell, 8-cell, then morula, blastocyst, day 5, and hatching and implantation on, like it is between the day 6 to day 9. Here we can see hatching and implantation. So this is how the things are going on. Procaudal plate. Around 14th day of development at the embryonic disc, the cuboidal cells of the ectodermal become columnar. This area is called procaudal plate. It later forms the endodermal layer of oropharyngeal membrane. It determines the central axis of the embryo and also enables us to distinguish its future head and tail end. Primitive streak. Around third week of development, some of the ectodermal cells lying along the central axis near the tail end of the disc begin to proliferate and form an elevation that bulges into amniotic cavity. This elevation is called the primitive streak. Here we can see this is the head end, this is the tail end. This is the endoderm and this is the ectoderm, the upper one. This is the procaudal plate. Now over here the primitive streak starts appearing. This is the primitive streak, ectoderm, endoderm, and between these two the mesoderm starts developing. Mesoderm, the cells that proliferate in the region of primitive streak pass sideways, pushing themselves between the ectoderm and the endoderm and form the mesoderm. That is the third germ layer. Mesoderm migrate in all directions between the ectoderm and endoderm, excepting near oropharyngeal membrane anteriorly and cloacal membrane posteriorly. This is the amniotic cavity. This is the ectoderm, primitive streak. Here the mesoderm is proliferating and spreading. This is the endoderm. This area is endoderm. And then the yolk cell. This is the yolk sac. Notochord. The cells of the primitive streak proliferate and move cranially in the midline between the ectoderm and the endoderm, reaching up to the caudal margin of the procaudal plate. The cells undergo differentiation and form a solid rod called notochord, which lies in the midline in the position to be later occupied by the vertebral column. However, the notochord does not give rise to vertebral column. The notochord terminates anteriorly at procaudal plate and marks the site of future pituitary gland. Most of the notochord disappears, but parts of it persists in the region of each intervertebral disc as the nucleus pulposus. Here we can see procaudal plate, notochord. This is the primitive streak, this is the cloacal membrane. The ectoderm, then the primitive streak, this is the cloacal membrane, notochord, and this is the endoderm. Derivatives of the three germ layers. We have already discussed about the ectoderm, endoderm, and the mesoderm. So there are three germ layers. So what are the derivatives of those three germ layers? From the ectoderm, skin and appendages, they are forming central and peripheral nervous system, oral mucous membrane, and enamel. From the mesoderm, cardiovascular system, heart and the blood vessel, locomotor system, bones and the muscles, connective tissues, pulp, dentine, cementum, periodontal ligament, and the alveolar bone. From the endoderm, lining epithelium of the alimentary canal, secretory cells of liver and pancreas, and lining epithelium of the respiratory system develops. Neural tube. The ectoderm over the notochordal process becomes thickened to form the neural plate, which is depressed along the midline to form the neural groove. The edges of the groove eventually fuse, converting the neural groove into the neural tube. Like over here we can see this is the neural groove and then the neural tube is formed. Neural crest cells. At the junction between the neural tube and the surface cutaneous ectoderm on either side, groups of the cells appear. They are known as neural crest cells. They are ectodermal in origin but have the properties of mesoderm, hence they are known as ectomesenchyme. We are talking about the neural crest cells. They are capable of migrating along the cleavage planes between mesoderm, ectoderm, and endoderm. After reaching their destination, they undergo cytodifferentiation. Several important structures are derived from neural crest cells. They are the neurons of the sensory ganglia of the 5th, 7th, 8th, 9th, and 10th cranial nerves, the neurons of the sympathetic ganglia, and the Schwann cells. The specific cells of the adrenal medulla and chromaffin tissue, melanocytes, calcitonin secreting cells, cartilages of the branchial arches, facial processes, all the tissues of the tooth except enamel and its supporting structures. Now, subdivisions of the mesoderm. Pharyngeal mesoderm. It is the mesoderm on either side of the notochord. It becomes segmented to form a number of somites that lie on either side of the developing neural tube. 42 to 44 paired somites appear in craniocaudal direction, of which there are 4 occipital, 8 cervical, then 12 thoracic, 5 lumbar, 5 sacral, and 8 to 10 coccygeal. These are somites. The pharyngeal mesoderm we are discussing, the somite is divisible into 3 parts. The ventromedial part is called the sclerotome. It gives rise to vertebral column and ribs. The lateral part is called the dermatome. The cells of this part gives rise to dermis of the skin and subcutaneous tissue. The intermediate part is myotome. It gives rise to striated muscle in the cervical, thoracic, lumbar, and sacral regions. One spinal nerve innervates each myotome. In somite period, cardiovascular system, alimentary system, nervous system, primordium of eye, and internal ear develop. Lateral plate mesoderm. It is laterally placed and gives rise to pleural, pericardial, and peritoneal cavities. Then connective tissues, muscles of viscera, blood, lymph cells, then cardiovascular and lymphatic system, spleen, and the adrenal cortex. Intermediate mesoderm, it gives rise to urogenital system. Folding of embryo. At this stage, the embryonic disc undergoes lateral and craniocaudal folds. With the formation of the head and tail folds, parts of the yolk sac become enclosed within embryo and a tube lined by endoderm is formed. This is primitive gut, which is in direct communication with yolk sac. The part of the gut cranial to this communication is foregut. The part of the gut caudal to this communication is hindgut. And the intervening part is called midgut. The whole of the gut is sealed off anteriorly by the oropharyngeal membrane and posteriorly by the cloacal membrane. Later, it is converted into a canal by breakdown of oropharyngeal and cloacal membranes. Derivatives of foregut endoderm. Laryngeotracheal diverticulum. They gave bronchi and lungs. Hepatic and pancreatic diverticulum, they gave liver and pancreas. Then pharynx and pouches, esophagus, stomach, first part of the duodenum. Derivatives of the midgut endoderm, rest of the duodenum, entire small intestine, ascending colon, transverse colon of large intestine. Derivatives of the hindgut endoderm, descending colon and rest of alimentary canal. Early orofacial development. Well, this early orofacial development, the cranial end of the embryo develops rapidly compared to caudal end. Stomatodium, the primitive oral cavity, is a shallow depression surrounded by neural plate cranially and cardiac plate caudally. The buccopharyngeal membrane separates it from foregut. Stomatodium forms the topographical center of developing phase. In the early somite period, 21 to 31 days of embryo, 5 mesodermal elevations augmented by neural crest cells and lined by ectoderm appear surrounding the stomatodium. They are facial processes and constitute the initial features of face. Branchial or pharyngeal arches. Well, during the late somite period, that is 4th week intrauterine, a series of distinct bilateral mesenchymal swellings augmented by the neural crest tissues develop on the ventral aspect of the embryo, just caudal to the head fold in the future mandibulocervical region. These sets of 5 or 6 elevations are known as branchial arches. They are separated by 4 branchial grooves on the external aspect of the embryo. Internally, they are separated by the 5 out pocketings of the elongated pharynx of the foregut, which we call pharyngeal pouches. Here we can see formation of the branchial arches and branchial grooves and pharyngeal pouches. So they start forming. This is the buccopharyngeal membrane, ectoderm, mesoderm, endoderm, and this is the primitive pharynx, the foregut. So the arches, they are appearing, and the grooves, and then pouches. The branchial arches decrease serially in size from the 1st to the 6th, each pair merging mid-ventrally to form semicircular collars around the pharynx. Only 5 pairs of arches are discernible, however, the 5th being transitory. In each pair of arches, there are a central cartilage rod that forms the skeleton of the arch and are differentiated from the neural crest tissue. A vascular component originates from the lateral plate mesoderm and a nervous element consisting of the sensory special visceral motor fibers of one or more cranial nerves supplying the mucosa and branchial muscle arising from that arch. A muscular component, the branchiomeric muscle component, are of lateral plate mesoderm origin. Now over here we can see pharyngeal arch derivatives. Pharyngeal arch 1, maxillary and mandibular. Nerve is trigeminal, which is the 5th cranial nerve. Artery, maxillary artery, terminal branches, neural crest, skeletal structures, mandible, maxilla, malleus and incus. Muscles which are originating from it, muscles of mastication, mylohyoid, tensor, tympanic, and anterior belly of the digastric, not the posterior, only anterior. And ligaments, anterior ligament of malleus and sphenomandibular ligament. From the second pharyngeal arch, which is called the hyoid arch, its nerve is facial nerve, which is 7th cranial nerve, and artery is stapedial, embryonic, during the embryonic period, and then the corticotympanic in the adult life. Neural crest, the skeletal structures, stapes, styloid process, lesser cornu of hyoid, upper part of the body of the hyoid bone. And muscles of facial expression, stapedius, stylohyoid, posterior belly of digastric muscle. So digastric muscles, anterior belly is coming from one arch, and the posterior belly of digastric is coming from the other arch. And stylohyoid ligament arises from second pharyngeal arch. Then comes the third arch. Its nerve is glossopharyngeal. Artery is common carotid, internal carotid arteries, neural crest or skeletal structures, greater cornu of hyoid, lower part of the body of the hyoid bone. Lesser cornu is coming from the second arch, and muscle is stylopharyngeus muscle. Then fourth one, vagus nerve, superior laryngeal branch of the vagus nerve is the nerve for 4th arch. Artery, part of the aortic arch left and part of the right subclavian artery on the right side. Then the skeletal structures, thyroid, cricoid, arytenoid, corniculate and cuneiform cartilages, they are arising from here. Then comes the muscles, cricothyroid, soft palate, levator, veli palatini, not the tensor veli palatini, only the levator veli palatini. Then the 6th arch, 5th As I told you, it appears and it disappears. Vagus, its branch recurrent laryngeal, is the nerve for sixth arch. Artery, part of the left pulmonary artery on the left side and part of the right pulmonary artery on the right side. The skeletal structures, thyroid, cricoid, arytenoid, corniculate, and cuneiform cartilages and muscles, larynx, intrinsic muscles, not the cricothyroid muscle. The branchial grooves and the pharyngeal pouches. After the formation of branchial arches between primitive stomatodeum and developing heart, they are marked on outside by a series of grooves lined by surface ectoderm and are known as branchial grooves. On the inner aspect of the pharyngeal wall are corresponding small depression lined by foregut endoderm called pharyngeal pouches that separate each of the branchial arches internally. The first branchial groove persists and deepens to form the external acoustic meatus, the ectomesoendodermal membrane in the depth of the groove. Separating it from the first pharyngeal pouch persists as the tympanic membrane. The second, third, and fourth branchial grooves become obliterated by the caudal overgrowth of the second branchial arch, which provides a smooth contour to the neck. Failure to obliterate completely these branchial grooves result in a branchial fistula leading from the pharynx to the outside of a branchial sinus or cyst, forming a closed sac. Pharyngeal pouches. The 5 pairs of pharyngeal pouches on the sides of the pharyngeal foregut form dorsal and ventral pockets, of which endodermal epithelium differentiates into a variety of structures. Elongation of the third, fourth, and fifth pharyngeal pouches during the 6th and 7th weeks intrauterine increasingly dissociates the pouches from the pharynx and allows their derivatives to form in the lower anterior neck region. First pharyngeal pouch. The ventral portion of this pouch is obliterated by the developing tongue. The dorsal diverticulum deepens laterally as the tubotympanic recesses to form the auditory tube, widening at its end into the tympanum or middle ear cavity, separated from the first branchial groove by the tympanic membrane. The tympanum becomes occupied by the dorsal ends of the cartilages of the first and second branchial arches that later develop into ear ossicles. The tympanum maintains contact with the pharynx via the auditory tube throughout life. Second pharyngeal pouch. The ventral portion of this pouch is obliterated by the developing tongue. The dorsal portion forms palatine tonsil and the tonsillar fossa. Third pharyngeal pouch. The ventral diverticulum endoderm proliferates and migrates from each side to form single median thymus gland. The dorsal diverticulum endoderm differentiates and migrates caudally to form the inferior parathyroid gland. Then comes the fourth pharyngeal pouch. The fate of endoderm of the ventral diverticulum is uncertain. The lining membrane may possibly contribute to thymus or thyroid tissue. The dorsal diverticulum endoderm differentiates into the superior parathyroid gland, which after losing contact with the pharynx migrates caudally with thyroid gland. The fifth pharyngeal pouch. The fifth pouch appears as a diverticulum of the fourth pouch, the endoderm of which forms the ultimobranchial body. The calcitonin secreting cells of the ultimobranchial body are derived from neural crest tissue and are eventually incorporated into the thyroid gland. Here we can see fate of the branchial groove and pharyngeal pouches. This is for the grooves. The groove 1, these are the pouches. Then they are obliterated second, third, and fourth because of the arch, second arch. And here pouches, external acoustic meatus, auditory tube. This is the second arch which is obliterating the grooves. Inferior parathyroid gland is developing here, and then the superior parathyroid gland and the ultimobranchial body. It is developing over here in the sixth pouch. Neural crest cell derivatives. Cranial Neural crest derived mesenchymal cells, ectomesenchyme, migrate into the pharyngeal arches from midbrain and hindbrain region, forming neurons, Schwann cells, smooth muscle cells, osteoblasts, chondrocytes, and odontoblasts. We have already discussed about it. Odontoblasts are coming from the neural crest cells from the ectomesenchyme. Development of tongue. Well, the tongue begins to develop at about 4th week of intrauterine life in the ventral wall of the primitive oropharynx in relation to first 4 branchial arches. Paired lateral proliferation of mesenchyme appear on the internal aspect of the first branchial arch to form lingual swellings. Between and behind the lingual swellings, there appears a median eminence, the tuberculum impar. Its caudal border is marked by a blind pit, the foramen cecum, from which the epithelium proliferates to form a downgrowth thyroglossal duct from which the thyroid gland develops. Another midline swelling appears in relation to the medial ends of the 2nd, 3rd, and 4th arches. This swelling is called the hypobranchial eminence, which soon shows a subdivision into a cranial part related to the 2nd and 3rd arches, which we call copula, and a caudal part related to the 4th arch. The caudal part forms the epiglottis. The anterior 2/3 of the tongue is formed by the fusion of tuberculum impar, the 2 lingual swellings. The anterior 2/3 of the tongue is thus derived from the mandibular arch. The posterior 1/3 of the tongue is formed from the cranial part of the hypobranchial eminence, which we call copula. The posterior 1/3 of the tongue is thus formed by 3rd arch mesoderm. The posterior most part of the tongue is derived from the 4th arch. Nerve supply of the tongue. A V-shaped sulcus terminalis whose apex is at the foramen cecum. It demarcates the anterior 2/3 and the posterior 1/3 of the tongue. According to the embryological origin, the anterior 2/3 of the tongue is supplied by the lingual branch of the mandibular nerve, which is the post-traumatic nerve of the 1st arch, and by the chorda tympani, which is pre-traumatic nerve of this arch. The posterior 1/3 of the tongue is supplied by the glossopharyngeal nerve, which is the nerve of the 3rd arch. The most posterior part of the tongue is supplied by the superior laryngeal nerve, which is the nerve of the 4th arch. Musculature of the tongue. The musculature of the tongue is derived from the occipital myotomes and are supplied by the hypoglossal nerve, which is the nerve of these myotomes. Epithelium of the tongue. The epithelium of the tongue is at first made by the single layer of cells. Later it becomes stratified. Stratified means having multiple layers, and papillae become evident. The line of the sulcus terminalis is marked by 8 to 12 valate papillae, which were previously known as circumvallate papillae, but now we call those valate papillae, that develop at 2 to 5 months of the intrauterine life. The mucosa of the dorsal surface of the body of the tongue develops filiform and fungiform papillae at 11 weeks intrauterine. At birth, the root mucosa becomes pitted by deep crypts that develop into lingual tonsil, and the completion of which is marked by lymphocytic infiltration. Taste buds arise by inductive interaction between epithelial cells and nerve cells from the chorda tympani, glossopharyngeal, and the vagus nerves. Development of human face. Well, human face, it starts with the appearance of facial primordia, which appears early in the 4th week around the primordial stomodium or stomatodium. That's how it is. The maxillary process, mandibular process. Well, actually there are 5 processes. The frontonasal process, 2 bilateral maxillary processes, and 2 bilateral mandibular processes around the future oral cavity, and they are appearing on top of the pericardial bulge. This is the frontonasal. then we have maxillary, and then we have mandibular processes. Five facial primordia appear as prominences around the stomodium. The single frontonasal prominence, which I told you, then the two paired maxillary prominences and the two paired mandibular prominences. The paired facial prominences, those are the maxillary and the mandibular, they are developed from the first pair of the pharyngeal arches. The prominences are produced mainly by the proliferation of the neural crest cells. These cells migrate from the mesencephalon and rhombencephalon regions of the neural folds into the arches during the 4th week. We have already discussed about that. So, first arches, they are, the first arch, they are forming the bilaterally maxillary as well as mandibular processes. The nasal placodes, these are the two bilateral oval thickenings in the surface ectoderm of the frontonasal prominence. They appear by the end of the 4th week. Nasal placodes are the primordia of the nose and the nasal cavities. Initially, these placodes are convex, means they are protruding outside. Later, they are stretched to produce a flat depression in each placode. Here we can see that. These are the nasal placodes. Nasal pit. Here we have lateral nasal prominence, medial nasal prominence. These are the mandibular prominences. Mesenchyme in the margins of the placodes, they proliferate, producing horseshoe-shaped elevations, which we call medial and lateral nasal prominences, which I have already showed to you. These are the prominences, medial and lateral. Now the nasal placodes lie in depression called nasal pits. These are the nasal pits, which were placodes. Now they are converted into the pits. These pits are the primordia of the anterior nares or nostrils and the nasal cavities. Mesenchymal cells are the major source of the connective tissue components, including muscles, cartilage, bone, and ligaments in the facial and in the oral regions. The frontonasal prominence is formed of two parts. The frontal part forms the forehead, and the nasal part forms the rostral boundary of the stomodium. The paired maxillary prominences form the lateral boundaries of the stomodium. And the paired mandibular prominences, they constitute the caudal boundary of the primitive mouth. So, rostral, that is the top surface coming from the nasal part of the frontonasal prominence, and the boundary which is caudal or at the bottom, that is being made by union of the two mandibular prominences. Facial development occurs mainly between the 4th and the 8th week. By the end of the embryonic period, that is 8th week, the face has an unquestionable human appearance. Between the 7th and 8th weeks, the two medial nasal prominences merge with each other and with the maxillary and lateral nasal prominences. Merging of the medial nasal and maxillary prominences results in the continuity of the upper jaw and the upper lip and separation of the nasal pits from the stomodium. Hence, the premaxilla is formed. The lower jaw and lower lips are the first parts of the face to form. Keep this thing in mind. The lower jaw and lower lips are the first parts of the face to form. They result from merging of the medial ends of the mandibular prominences in the median plane. median cleft lower lip is a very rare condition, but it occurs. Each lateral nasal prominence is separated from the maxillary prominence by a cleft called nasolacrimal groove. This area. If we look at the schematic diagram, we can see it over here, nasolacrimal groove. Development of the palate. As the medial nasal prominences merge, they form an intermaxillary segment. The intermaxillary segment gives rise to the philtrum, the median part of the upper lip, the premaxillary part of the maxilla, and associated gingivae, the primary palate. This part which is in yellow. This is the philtrum area, then maxillary 4 incisors, and the palate, anterior palate, the primary palate. Development of the palate we are discussing. The palate develops in 2 stages, the primary palate and the secondary palate. Palatogenesis begins at the end of the 5th week and is completed by 12th week. The critical period of the palate development is from the end of the 6th week until the beginning of the 9th week. This is the time when clefting could occur. Primary palate. Early in the 6th week, the primary palate or median palatine process begins to develop from the intermaxillary segment of the maxilla. Initially, this segment is formed by merging of the medial nasal prominences. The primary palate forms the premaxillary part of the maxilla. It represents only a small part of the adult hard palate. Two medial nasal prominences, they merge and they make the intermaxillary segment and gives rise to the primary palate. Here we can see 2 medial nasal prominences. When they merge, they give the primary palate. Secondary palate. Well, the secondary palate is the primordium of the hard and soft palate. It begins to develop early in the 6th week from 2 mesenchymal projections that extend from the internal aspects of the maxillary prominences. Here we can see the palatal. The tongue, before it regresses, the tongue is lying here. And if it does not regresses, then the merger of these palatal shelf is not going to occur. Remember this thing. Initially, the lateral palatine processes or palatal shelves project inferomedially along the tongue on each side of the developing tongue. And then the tongue regresses, as I told you, and it provides the support or scaffolding to the developing secondary palate, and these palatal shelves, they fuse with each other as well as with the nasal septum. Like this. As the jaws develop, the tongue becomes relatively smaller, as I told you, and moves inferiorly. During the 7th and 8th weeks, the lateral palatine processes elongate and ascend to a horizontal position superior to the tongue. Gradually, these processes approach each other and fuse in the median plane. Well, palatine processes also fuse with the nasal septum, as I told you, and the posterior part of the primary palate. The nasal septum develops as a downgrowth from internal parts of the merged medial nasal prominences. The fusion between the nasal septum and the palatine processes begins anteriorly during the 9th week and is completed posteriorly by the 12th week. If tongue does not regress, then merger is not going to occur, or fusion. Here, palatal shelves move medially and fuse with the nasal septum as well as fuse with each other. Well, Cone gradually develops into the primary palate, forming the premaxillary part of the maxilla which lodges the incisor teeth. Concurrently, bone extends from the maxillae and palatine bones into the lateral palatine process to form the hard palate. The posterior part of these processes do not ossify. They extend posteriorly beyond the nasal septum and fuse to form the soft palate and then further to make the musculus uvulus. Its soft conical projection, we call it uvula. Well, if they are not merging in this area, we can have even bifid uvula as well. The median palatine raphe indicates the line of fusion of the lateral palatine processes. If we look at an adult palate, we can see the premaxillary part with the incisive fossa, then we have the intermaxillary suture or interpalatine suture, or then we have the palatine bones, and we can see the palatine processes of maxilla. Cleft lip and palate. The defect is usually classified according to the developmental criteria. There are two major groups of cleft lip and palate. Clefts involving the upper lip and anterior part of the maxilla, clefts involving the hard and soft regions of the palate. Here we can see different types of clefts. It could be just the cleft of the lip, it could be cleft lip and palate, it could be unilateral, it could be bilateral, it could be just the clefting of the anterior part, that is the clefting between the primary palate and the secondary palate, or it could be a through and through clefting. Then other than that, we can have oblique facial clefts, bilateral hair lips, median hair lip, macrostomia, unilateral macrostomia, and microstomia. Bifid uvula, as I told you, it could also occur unilateral hair lip with incomplete cleft. Then we have unilateral hair lip with complete cleft palate, and bilateral hair lip with bilateral incomplete cleft palate. Yes, even bilateral hair lip with complete cleft palate can also occur, and it is quite an extensive defect. Now, quick review. Development of the face and palate, weeks 4 through 10. The groove between the maxillary prominence and the lateral nasal process is called the nasolacrimal groove. This is the nasolacrimal groove. Medial nasal process, lateral nasal process, maxillary prominence. What if they don't fuse? We will be having oblique facial cleft. Oblique facial cleft and oblique facial cleft occurs when the lateral nasal prominence and the maxillary prominence fail to fuse. Oblique facial clefts result in the nasolacrimal ducts becoming open grooves. Nasolacrimal groove, intermaxillary segment, lateral nasal processes, and the maxillary process. We can see these are all identified by these colors. Then development of the face and palate we are discussing. During the 7th week, the two medial nasal processes fuse to form the intermaxillary segment. What if they don't fuse? Then we are going to have median cleft lip. Here, using these colors, we can identify these intermaxillary segment, lateral nasal processes, and the maxillary prominences. Median cleft lip. This abnormality is rare and usually associated with the other problems such as mental retardation or brain abnormalities. The intermaxillary segment and the maxillary prominences fuse, forming the upper lip and jaw architecture. The intermaxillary segment and the maxillary prominences fuse, forming the upper lip and the jaw architecture. What if they don't fuse? We are going to have cleft lip. Cleft lip. Cleft lip, the failure of intermaxillary segment and the maxillary prominence or to fuse occur in adult 1 per 1000 births and is much more common among the males. Cleft lip may be unilateral on one side only or bilateral on both the sides and may appear as a slight indentation on the lip or extend as a groove up to the nostril. There are many potential causes and risk factors for cleft lip. By 10th week, the facial features resemble that of a neonate. The intermaxillary process forms the philtrum of the upper lip, and at this point, maxillary prominences are starting to form well-defined cheeks and portions of the upper jaw. Pharyngeal arches and derivatives, just the quick review. Pharyngeal arch 1, mandibular arch has two prominences. Smaller upper maxillary forms maxilla, zygomatic bone, and squamous part of the temporal. Larger lower mandibular forms the mandible. Pharyngeal arch 2, hyoid arch forms most of the hyoid bone. Arch 3 and form neck structures. Arch 6, thyroid and cricoid cartilages and intrinsic muscles of larynx. So over here we are done with this lecture, and hopefully you have enjoyed it. I tried to make it as comprehensive as possible for all of you.